So good morning, everybody. My name is Simón, and I am the Scientific Director at BeeLife, and I am going to be your moderator of this session. That is a scientific conference where we are going to learn more about European projects dealing with pollinators, monitoring trends, threats, risk assessment. It's going to be very, very intense in information, but we are going to learn a lot of the nice... Sorry, we have a technical problem. Let's see. So now we can continue. Excuse me, as we have some technical issues. So I was saying that we are going to have, it's a session that is going to be very intense in information, and we are going to see the richness of European research projects, and if we could say administrative research projects. And and we are going to learn a lot about the actions and maybe some of the results that some of these projects have had so far. Some household rules, please, for the attendees. If you have any questions to the speakers, you can write them in the Q&A, okay, and we will check it. So each of the speakers are going to present, and then we have time, very short time, for one question, two questions. It is very fast. And hopefully, maybe at the end, we will see, we will have some more time for questions. Anyway, if you still have not responded questions that you would like to be addressed, please, well, you can address them to them, because I imagine each of the speakers have included their contact details in their presentations. So, and with this, I would like to give you Ignasi Bartomeus, or Nacho Bartomeus, who is going to speak from Spain, from the CSIC in the south of Spain. And he's an expert in ecology of wild pollinators, and he's going to present us some news about the Safeguard project and some of the results that they got there. So, Nacho, the floor is yours.
Thank you very much. Let me put my presentation. So it's a pleasure to be speaking here today. And today I would like to start this scientific session on pollinators by going through a topic that I think it's really important, which is that it's really difficult to protect something that we don't know it exists. And despite we know quite a lot about bees and pollinators, there's still big knowledge gaps that we need to fill. And maybe some of those will surprise you, but in the European Union, we have more than 2,000 bee species. And in the last EUCN assessment, we can see that we have very few information for most of those. In the panel in yellow, you can see the richness of different countries and regions in Europe, and you can see the southern and Mediterranean countries have a large richness. But more than 50% of the species, we don't have enough data to know if they are doing okay or if they are doing bad. This is for me the most striking number that I get from this threat list exercise. That means that we need to know better these basic things about the status and trends. We do know some things. For example, there is this classic paper analyzing some sites in Britain and in the Netherlands, where they compare historical data with more recent data. And we can see changes, for example, in the species richness. And as you can see in the top panels for bees, there is a lot of red. Red means that the current observed richness is lower than the historical one. Sylphids are doing a bit better. We can see a bit of blue, both in Britain and in the Netherlands. But despite these indications of declines, I would like to highlight that the documented extinctions on pollinators are luckily very few. For example, this paper by Jeff Ollerton showed that there is less than 20 recorded hymenopteran extinctions in the British Islands historically, which this is good news. We may have some declining trends, but we are still in a moment where extinctions are not occurring, at least at a large range. But these two areas, both the sites near London and the Netherlands, are representative of what's happening in the rest of Europe. Well, we don't really know because we lack data from most of these areas. That's why Safeguard, in one of the main tasks, is helping update this UCN red list. And one thing that we are doing, for example, is collecting and digitalizing records from Spain. We have more than 1,000 bee records already from Spain, historical records and recent records. And this source of information is really rich. What can we do with this kind of information? I'm gonna use 3 examples, all related to the Iberian Peninsula, to illustrate which kind of information we can leverage from this rich information. First of all, I want to put an example on how can we track also climate change effects. This is because Europa in the north of the Iberian Peninsula, and we had very nice records from the lowlands almost at the sea level starting, and how bumblebee change across when we increase in altitude up to almost 2,000 meters. And the nice thing is that we have the historical records of this bumblebee occurrence, and we have the newly collected records. And we can see in this graph, if we go to the bottom, the places which are at low altitude, we are losing species. The net effect is that we have local extinctions. And this happens when we move in altitude, but at some point, we start gaining species. In the upper altitudes, we can see in the part of at the upper part of the graph, we can see local colonizations. So species that used to be in the lowlands are moving up to keep up with climate change. This is very interesting, both from the perspective of understanding changes in bee populations, but also for understanding the drivers of these changes, in that case, driven by the current climate change. Another example I wanted to show is in Castilla y León, where we also have historical data previous to 1980s, and that was really well collected data by Enrique Asensio, who was an entomologist who did very nice bee surveys. So we could go back to the same places and resurvey those. And what we found is a decrease in richness. So you can see most of these dots are lower in 2016, and some of them are quite worrying declines. But I would like to focus on these 2 dots in the bottom, like the red and the green one, because for these sites, we recovered almost 100% of the species that were present. And these 2 sites are the ones that are in the mountain slopes, as the one you see in the picture. So that means that we have an heterogeneity of responses. So some areas, like the ones that are suffering more urbanization or agricultural intensification, will be losing species, but in some others, we can be maintaining very nice species richness. And this is very encouraging because we know which kind of measures we can take to buffer these trends. The final example is an example from Doñana National Park, where I am working, when we can also see the effects of climate change. What we are seeing in these graphs is the activity of the bees along the season. And so the day 0 is the first day of the year. And we can see how we are seeing every year earlier phenology. And despite there are some variability, because not all years are equal, we are seeing an advance on this phenology, which still unknown consequences for the plant-pollinator interactions. So with this information in Safeguard, and not only for Spain, but for all Europe, we are trying to understand not only the status and trends, but also the drivers. that are causing these changes. In fact, a few work packages are based on generating evidence, which is very important, as I said, on the status and trends, the drivers and pressures, and which actions we can do to mitigate these declines, but also impact generation in terms of bringing tools and solutions, integrating policy, our results in policy and decision-making, and knowledge exchange. With this aim, SafeWater still have 2 more years to go, and we will leverage across Europe information from individuals to community level to populations and the functioning of pollinators. And that was the highlights I wanted to share. Thank you very much.
Thank you, Nacho. Nacho?
Yeah,
you're still there. Okay, because you were frozen. So is there any question? So I don't know if you finished, because at the end you were frozen.
Oh, sorry. Yep, I hope you listened most of it.
Yes. So it was only at the last time that you kind of yeah. So I don't know if there are any questions from the audience. I don't see anything in the Q&A. I think you were actually very didactic. Thank you very much. So since we don't have more anyway, please don't hesitate to write in the Q&A, and then Nacho is able to answer in writing. So thank you. With this, I'm going to we pass the floor to David Roy, who is going to talk from the UK. We actually have a very good representation of the European in this panel. David is an expert in environmental monitoring and assessment, and he has been involved into many monitoring of pollinators, and among them the spring, that is actually the strengthening pollinator recovery through indicators and monitoring. And it's a current project, and we are very eager to know more about this project. The floor is yours, David.
Great, thank you very much, and thanks for the invitation to speak today. I'm going to present an update on behalf of the Spring project, which is finishing in a couple of months. So we're busy processing the data and collecting in the information to allow us to report more fully. So I'm really going to give an overview today of the project and some of the I'm trying to illustrate the project through some highlights and numbers from what activity we've done so far. And by way of background, I guess this diagram may be familiar to many of you. It comes from a very extensive report led by Simon Potts that involved a large number of experts across Europe to review options for how a EU pollinator monitoring scheme may work. This diagram illustrates the pillars of activity that would be necessary for a complete scheme, and it covers the different sampling methods, different taxa that we're targeting, so particularly focused on bumblebees, hoverflies and butterflies, but also recognizing other pollinating insect groups such as moths and wider biodiversity. So, it also considers the participants in terms of people recording in the field, the balance between volunteers and professionals, and to deliver a range of metrics of the status of pollinating insects, species abundance, diversity, etc. And it's really, as Nacho clearly illustrated, we need evidence to make decisions about and to understand how pollinators are faring and what the drivers of change are. So the Spring project is taking this diagram and the recommendations of the Potts et al. Report. There's a link there. I encourage you to read it. It's a lot of very detailed and excellent information. And the Spring project is piloting some of the recommendations in terms of field methods, capacity building, etc. And the red squares on this diagram illustrates part of the overall program for pollinators that we are piloting and testing in the Spring project. So I'm going to, as I say, I'm going to illustrate some of the activities of Spring in terms of numbers. So my first number is 27, which I'm sure you know is the number of EU member states. And we are very pleased that as part of the Spring project, building on a previous project called ABLE, working particularly with butterfly conservation and butterfly enthusiasts around Europe, we've now established a butterfly monitoring scheme through a citizen science approach in all EU member states. Clearly, there's work to sustain this effort, but the establishing the scheme and the coordination mechanism has been set up by the Spring project. So this is building on some long-established monitoring schemes in the Netherlands and Catalonia, for example. But as I say, now having some activity in all EU member states. And this is important because the butterfly monitoring data delivers the only insect-based monitoring for the EU, delivering the European Butterfly Index indicator that's used by the European Environment Agency as one of its indicator set. And the figure below shows us reporting a 30% decline in butterfly populations, this set of grass and butterfly species over the time series for the EU. And when we add in more data from other European countries outside the EU, the decline is slightly more. So that's a clear example of citizen science data delivering some baseline information on the status of pollinators. We've also been looking at other options for pollinator citizen science approaches. And our numbers here are as we ran a survey across Europe and also input from wider afield across the world to understand the citizen science activities that were being undertaken already, and to look at the barriers and opportunities around citizen science to adopt approaches for more pollinating insect groups through citizen science monitoring. So we're busy writing this up for our report and for publication. But we had a great response to this survey and have clear messages about some of the barriers and opportunities for citizen science. And one of the elements of citizen science that we have progressed under the Spring project is building upon one method, which is the flower insect time count method, which is a simple method to count insects visiting flowers and not requiring a high level of taxonomic expertise. So recording is to broad groups of pollinating insects. And this has proved effective at engaging new audiences in pollinator monitoring and also delivering valuable data on insect numbers, but also how they are associated with particular flowering species. So this mobile application has been adapted for 8 EU countries now and has also been used in South America. So that's the sort of citizen science elements of the Spring project. Another big part of the project was capacity building. So our next number is 42, which, although as well as being the ultimate question of life, the universe and everything, is the number of Spring courses that we've run across the EU over the last 2 years. Some of the diagrams here show courses run in the lab in terms of identification of insects and using keys, etc. Also explaining how to monitor populations, including field aspects. And this has been a great activity, both aimed at beginner level contributors, but also some very expert taxonomists to upskill the community in identifying some very tricky groups of pollinating insects. And 42 courses have had over 450 participants from 30 different countries. And in terms of evaluation, a great score of 9 out of 10 for those participating as to their satisfaction of the course material. And we brought all this material together. This has been led by the Naturalis Museum in the Netherlands to develop a Pollinator Academy, which is a home for all the resources that we've developed to provide training, facilities and taxonomic keys, species information, fact sheets, etc., to really build capacity across Europe that will clearly be needed to deliver a European pollinator monitoring scheme at the scale that will be required. The next number is 270, which is our field piloting of the main minimum viable scheme methods that were recommended through the Potts et al. Report. And so we've been piloting and field testing 2 primary methods. One is a transect-based method, where you walk a fixed route and count insects encountered and collect those that need identification in the lab. And then the second method is water traps or pan traps, illustrated here by these 3 bowls, which is a method where the traps are left out for a period of time during the day. Insects land in the liquid and then are identified separately through a lab process. And these 2 methods are complementary in terms of giving information on the diversity of pollinators or the abundance of pollinators, and also complementary in terms of the species that are best detected by the 2 methods. So in terms of the transects, we've had a great involvement with volunteers who've been encouraged to be involved in the scheme and have walked a huge distance in terms of sampling pollinating insects across Europe, and have counted huge numbers of bees and hoverflies and butterflies. And we're pulling this data together to understand how it can contribute to a monitoring scheme going forward. And our most commonly encountered species, particularly from the butterflies, is the meadow brown, which is a hugely abundant species across Europe and regularly encountered. In terms of the pollinator data captured, again, a huge effort from volunteers, over 650 field days and a large number of hours in the field. This collects a huge amount of material, so we're busy, or the teams are busy identifying and sorting this material and entering the data so that we can report across the project. But already we've have data for over 140 species of hoverflies and over 330 species of bees, and a large number of insects sorted in the lab. So huge effort from all those teams. And in terms of our most commonly identified or captured bee species is this Blasiogosum species, and for the hoverflies, this Heliophilus species. So we're collecting both abundant species, but also a huge diversity of both bees and hoverflies, which is important to get an overall picture of the pollinator communities across Europe. We've also been doing work in more limited places, but we've been testing a method for monitoring moth species. There's increasing recognition that the importance of moths is complementing bees and hoverflies as pollination, particularly providing that service at night. And again, a large number of so the method is a simple trap-based method, but supported by a mobile application for entering the data and using image recognition to help with the identification of species. And a large number of individuals, and over 1,500 moth species, have been recorded through these methods. My final couple of slides. So the most important number really for me is the huge contribution of the partners that have helped deliver the Spring project. And each partner is then connecting to a huge body of volunteers and experts who've been supporting the project to deliver all the things we need to deliver. So huge thanks to the group of organizations here, but also the wider network that they represent. And finally, I've talked about the Spring project today, but Spring project doesn't sit in isolation. It's linking very closely to work of butterfly conservation for the butterfly monitoring, but also 2 other European funded projects to build up taxonomic resources for hoverflies, the Taxofly project, and for bees, the Orbit project. So giving us some vital information for capacity building for those taxonomic groups. And this collection of projects will produce recommendations that move forward in terms of a planned rollout of the scheme in 2026 as part of the Nature Restoration Law. And the STING group is a group of experts, again, who delivered the initial recommendations, but are continuing to work to review the recommendations and the science base to make final recommendations to member states as to how the scheme can be rolled out most effectively. So thank you very much.
Thank you, David. I mean, we must say that the last 2 slides, they froze. So actually, we were frozen in the moths, the amount of individuals that you. So
okay, yeah. I can see on my screen. Sorry.
Yeah, I mean, I don't know why. I think maybe there is an internet connection problem or something. I don't know. I actually wanted to profit to ask you one thing. In the applications that you have developed in the Spring project, how do they link, for instance, with other applications that are out there, like eNaturalist or the GBIF databases? Thank you.
Yeah, so we're busily preparing data to share through GBIF, for example. The main applications that we link to are the systems for monitoring butterflies across Europe as one system. We're also benefiting from a partnership with Naturalis and the observation.org system, which provides the image classification methods for moths that we're using in the systems that we're developing.
Thank you. So I don't see that there were any more question and answers, but please, audience, don't be shy. You can send a question. It was also a very didactic and very clear presentation. So yeah, I can imagine that maybe the people don't have, let's say, questions maybe about the methodologies and things like that you go much more in depth, let's say. But I think it was also very clear. Thank you, David. Our next speaker is, we are actually going to leave the wild pollinators, let's say, and the trends and let's say and monitoring, and we go more into the honeybees and beekeeping practices, because we have next Giovanni Formato. He is a veterinarian and he is the coordinator of the project B-THENET. That it is a thematic network about good or sustainable beekeeping practices, let's say. Giovanni works in Italy at the Istituto Zooprofilattico Sperimentale del Lazio and della Toscana, that I always have problems with saying this name. And he is also participating at the FAO, with the FAO, for setting up a number of expert groups and work for spreading good beekeeping practices. So with this presentation, Giovanni, I leave you the floor.
Thank you very much. So can you see the presentation, right? Okay, thank you. So first of all, I'd like to thank BeeLife and Noah in particular to invited us to present our contribution to this scientific conference that is dedicated to provide you an update about the B-THENET thematic network. It represents the first thematic network of beekeeping in Europe. It is a 4 years long project. We are finishing the first year of the project, and the goal of this thematic network is work all together, the community of beekeepers, of researchers, policymakers, and all other stakeholders in defining the best beekeeping practices and identifying the innovations that could be adopted in the European beekeeping. Everything is to improve the beekeeping sector and to improve the sustainability inside of the European Union and European beekeeping. So the thematic network is characterized by one side by the multi-actor approach that is at the end working together. And it is even characterized by a bottom-up methodology. Indeed, we start identifying the best beekeeping practices, starting by the beekeeper's need. Among the best beekeeping practices that we are identifying, let me tell you that even the positioning of the apiary has been addressed as one of the most important practices for beekeepers. And indeed, they have to place apiaries unfortunately, to be sure that their bee will not die, far from conventional agriculture. So we learn beekeepers how to prevent and how to even control damages that could be given to the apiaries because of the not proper use of phytotherapeutical products. In the same time, we develop best beekeeping practices that are oriented even in supporting beekeepers on properly controlled diseases and even on general rules that are useful for bee health and good beekeeping practices. As you can see by this image, we have 14 countries that are involved in this project. The partners of this project are 18. We are happy to involve first of all beekeepers at the international level, like BeeLife, like Apimondia, like several other beekeeping associations from all over Europe. And together with the beekeeper associations, we work together with the universities, research centers, and centers that are specialized for social sciences, communication. This is to make easier to transmit information about best beekeeping practices to beekeepers and advisors in Europe. So this is the structure of the thematic network. We start in identifying the practices according to their relevance by the European beekeepers' point of view. Then we pass to exchange activities in which beekeepers are able to discuss with researchers in setting up different practices and adapting the practices according to different beekeeping realities in different European countries, and even considering the different kind of use that they have in their countries, even considering the different climatic situation of different countries. Once that the practices have been finalized and they are validated by the different countries, they are placed in a repository where they become accessible to all beekeepers for training and of course even for educational realities for certifying training of beekeepers. Here is our website. If you write down www.b-THENET.eu, you can open the website. You can find a part of the website that is dedicated to the project, and another one that is dedicated to the three platforms that we have seen. So the first one is to prioritize practices. The second one is about the exchange of practices, and the third one is about the repository where practices are placed for educational activities. Here is what you will see. You could see practices that are organized into different categories. One is about general good beekeeping practices. Another one is about biosecurity measures that are focused on how to prevent and control diseases and intoxication. So as we said, there are 13 countries involved in this project. In each country, we have physical centers where beekeepers can meet and can have access to beehives and test practices together with a moderator that is from the beneficiary of that country, of this project. And we have contact in this way with the beekeepers, advisors, and other stakeholders that register on the platform and are interested to be trained on practices and are interested to have exchange about different beekeeping practices in Europe. In the same way, we have 3 international B-THENET centers. Again, these are physical centers that have the task to have an international approach to the best beekeeping practices. This is why they provide practices written in English. On the contrary, at the national level, practices are written in the local language, local national language, so the beekeepers can participate to the exchange and can easily read and take knowledge by the practices that are described. So as international B-THENET centers, we have International Center for Beekeepers that is located in Italy and is managed by Apimondia. We have two international centers for advisors. So we have in the University of Sweden, the International Center for technicians, expert technicians in beekeeping, and in University of Madrid, University Complutense of Madrid, we have the International Center that is dedicated to training of veterinarians in Europe. Finally, we have the International Center for all other stakeholders. So these will include policymakers and all other actors that are different by beekeepers and advisors in Europe. It is coordinated by BeeLife, and just staying in contact, just continuing on this topic about the international centers. So here you can see some training activities and exchange activities on best beekeeping practices in different countries. And let me speak to you more in detail about an international event that is the B-THENET transnational event for all other stakeholders that was organized by BeeLife last July and in Romania. And one of the relevant output of that event was that in Europe, in some countries, beekeepers have difficult to access to the medicines to treat against Varroa. So, as probably most of you know, beekeepers, they stay away from chemicals that are used for agriculture because those chemicals can kill their bees. But on the other side, they are interested to medicines that are useful to treat Varroa mite, that is the most relevant disease in Europe. And of course, with this project, we try to orient beekeeping beekeepers toward the organic approach and using organic compound to treat their bees. But the problem is that in some countries, beekeepers have difficult to access the medicines. This is why we have a symposium, a satellite event this afternoon from 1:30 to 5, from 1:30 p.m. to 3, where we will discuss about the availability of veterinary medicinal products for beekeepers in Europe. So here you can see the schedule, and in the chat you can find the link if you are interested to participate to the forthcoming meeting that we will have. So I thank you very much for your attention.
Thank you. Thank you very much, Giovanni. So, well, we have people, I think it's important to say for all of the, thank you very much, Giovanni.
Thank you.
I wanted just to, so we have one question, and it is, so how can beekeepers and advisors from other countries that are not in the project have access to the practices and the assistance? Thank you.
Thank you. This is a very interesting question. Of course, beekeepers can take inspiration from beekeepers from countries that are not involved in this project can take inspiration from the, I think, especially by the activities of the international B-THENET centers, as they write down in English the practices, they describe the practices in 2 steps. The first is to describe in general the best beekeeping practice, and the second is to specify differences among countries. So I think that if other countries are interested to this project, first of all, we will be very happy to support them. And for example, now we received from Portugal the request to be part of this project, and we are trying to include them starting from the 2nd year of our project to integrate them. So first option could be let us know so we can try to include you in this project. The second possibility is that other countries can start giving a look to the best beekeeping practices that will be the output and will be included in the repository platform that is open access. And from there, they can start working and tailoring. This is a wording that Noah, for the first time, specified while we were writing the project, and it is very appropriate in my opinion. So different countries can start from our description at the international level, and then can go ahead specifying the reality in their country. And eventually we could even integrate those practices in our repository platforms, if everybody agrees on this. Thank you.
Thank you, Giovanni. So again, if other people want to have access to or want to have some other questions, please write them in the Q&A. So, okay, so I give, and in the chat, we have put, for those of you who want to participate to this afternoon B-THENET meeting on accessibility of veterinary medicines. It is, I mean, just
You're muted.
Sorry, I think I have been writing. I have been speaking and nobody could hear me. So thank you very much, Giovanni. I just wanted to say that in the chat, you have everybody the link to participate to this afternoon event, B-THENET event on accessibility to veterinary medicines in beekeeping, that it is not from 1 to 3:30, but from 1:30 to 3. Yeah, it's so it's okay. And then yeah, with this we are going to give the floor to Jordi Bosch from the Center of Ecological Research and Forestry Applications in the Universitat Autònoma de Barcelona. Jordi is also an ecologist and an ecotoxicologist. And now, actually, we are going, this is a transition towards what I was introducing at the beginning, more the administrative science, if we can say, even if it is science. And there is a lot of fundamental and applied scientific work on the issue. So Jordi is going to present his work on the combined effect of pesticides and other stressors on solitary bees. And yeah, so I give you the floor, Jordi.
Thank you, Noah. Can you see my screen all right? Can you see my screen? Can you confirm you see my screen?
Yes, we see.
Oh, okay, thank you. So yeah, the title of my presentation is Combined Effects of Pesticides and Other Stressors on Solitary Bees. And I'm going to show you the results or part of the results of this stress bee project. It's an ongoing project funded by the Spanish Ministry of Science and Innovation. Okay, well, as we've heard in previous talks, bees are in decline, and one of the drivers of these declines is exposure to pesticides. Now, to to protect bees from pesticides, the companies that produce them have to undergo a risk assessment process. That means that if we apply these products following the instructions of the label, we should avoid incidents such as this one, these massive mortalities. However, even when we use these plant protection products according to the label, that doesn't mean that bees are completely safe from them. Every time we spray a crop, some residues remain in the environment. These residues can be found in the pollen, in the nectar, in the soil, in water, and in other matrices that bees use to build their nests and to provision them. For the most part, these residues are found at low concentrations, and by themselves, they should not pose a threat to bees. But the problem is bees are confronted with multiple compounds at the same time. So the key question is, what happens when bees are exposed to low concentrations of a variety of pesticides? And this is what we attempted to answer in this study, in this experiment. We work with this solitary bee, which is so very different from a honeybee. The biology is completely different. Well, this bee emerges in the spring, and then we expose it to combinations of pesticides. In this experiment, we had 6 groups of bees. Control group that was fed regular syrup, that means water with sugar. Then a second group that was fed the same syrup with a fungicide. 2 other groups were fed the syrup with very low concentrations of an insecticide. These are, I insist, they're very low concentrations, they're realistic concentrations. In fact, they have been measured in the nectar of oilseed rape and apple, respectively. And then the final 2 groups in green, we exposed bees to a mixture of the fungicide and the insecticide, always at the same concentrations. Okay, following this exposure phase, we mark the bees individually with number tags, and we release them in these sealed cages. In this way, we can follow the nesting activity of the bee, and we know how many eggs each female has laid. I'm going to show you the population growth curve of the first group of bees, the control bees, the ones that were fed syrup. We start here at when 100% of the bees that were released in the cages are alive, okay? Then we have a slight decline, population decline, because some of the bees are too weak to start nesting. But the ones that do nest start laying eggs, and then the population increases. And we have another population decline because some eggs and some larvae die during development. And then, as you can see, during wintering, we have almost no mortality. Now, if we look now at the curve of the population growth curve of the bees that were fed the fungicide, you see the 2 curves are almost identical. So this is good news. Our result agrees with the results of risk assessment, which tells us that this fungicide is non-toxic to bees. Okay, let's look at what happens with the bees that were fed the low concentrations of insecticides. Population, these are the population, the growth rate is in the control bees, and the fungicide bees is 1.7. That means we recovered a population the following year that's 1.7 times greater than the one we started with. With bees exposed to the insecticide, the growth rate is slightly lower, 1.4, but this is not significantly statistically significant. So these populations can be sustained at these low concentrations of insecticide. However, when we mix the 2 compounds together, we have a negative growth. We lose 40% of the population. What's happening is that the presence of the fungicide is enhancing the toxicity of the insecticide. We have 2 populational bottlenecks, one here when fewer bees establish, when they're fed the mixture, and the ones that do establish have a shorter lifespan and therefore lay fewer eggs. So clearly this population under these conditions is not sustainable over time. Well, but bees are not only exposed to pesticides, they're only exposed to other stressors. These include insufficient floral resources, nutritional stress, climate change, parasites, diseases, and others. In this second experiment, we asked the question, what happens when bees are exposed to 2 stressors in combination, in this case, climate change and pesticides? Again, we work with the same bee. It's the solitary bee. We have 3 groups of bees that we expose to 3 wintering scenarios. One is the current scenario. These are the temperatures that have been recorded in the area of origin of this population between 2007 and 2012. The near future scenario is the projected temperatures between 2021 and 2050, and the distant future between 2051 and 2080. And all this is based in the models of the IPCC climate change report. After winter, in spring, we expose these to three pesticide scenarios. One, again, is a control, water with syrup. The other one, a low insecticide dose. And the third group, slightly higher insecticide dose. As in the previous experiment, these are realistic exposures, levels of exposure that have been measured in crop plants. Then we submit the bees to this test. You have to imagine that this is a dark box. We can see it because it's being filmed with an infrared camera, but the only light source really is this point here. And bees show an innate preference, an innate tendency to work, to walk or to move towards light. This is a control bee, a bee that was fed syrup. You can see she walks to the light right away. This instead is a bee that was fed the sublethal insecticide concentration. She's alive, she can walk, but she really doesn't know where she's going. She keeps walking for a long time and does not reach the light source. The results of this experiment are summarized in this graph. Here you have the percentage of bees that responded to light. You have the three groups of bees, the current scenario, near future scenario, and the distant future scenario. And the white bars are the bees that were fed the controlled syrup. And you can see almost 100% of them responded positively to light. In the gray bars, with the bees that were fed the low dose, we start seeing a decline, but this is really non-significant. But with the higher insecticide dose, the percentage of bees, the fraction of bees that are responding to light really decreases very importantly and very dramatically in the bees of the distant future scenario. We also measure the longevity of these bees in the current scenario and the near future scenario. The bees that were fed the controlled syrup and the low dose of insecticide lived for about 14 days. Excuse me. Instead, the bees that were fed the high dose of the insecticide, they saw their longevity greatly reduced, almost by half. And in the bees of the distant future scenario, you can see that even the bees that were fed the regular syrup, they see their lifespan significantly reduced. And when they are exposed to a high dose, or to the higher dose of the insecticide, they only live for about 3 days. These bees lay about 1 egg per day, so it's impossible to sustain a population under these circumstances. Well, the conclusions of these experiments are various. First, some pesticide combinations have synergistic effects. Second, some realistic pesticide combinations cause sublethal effects, and these sublethal effects may hinder bee population growth. Third, synergistic interactions also occur between different drivers of bee declines. Very importantly, the wintering temperatures of our most pessimistic scenario are already occurring. We have already had winters as warm as the temperatures we simulated in the third scenario. So this is no longer a projection. This is already happening. Climate change is making bees more vulnerable to pesticides. These results have important implications for risk assessment. First, risk assessment should not only test lethal effects, but also sublethal effects. Second, risk assessment should not test just single products in isolation, but combinations of products. Number three, risk assessment should also test combinations of stressors. Number four, risk assessment should not only rely on the honeybee. As already proposed by EFSA 10 years ago, risk assessment should include solitary bees and bumblebees to get a more complete picture of what's happening with bee communities. Number five, risk assessment should emphasize chronic exposure. Bees are not exposed to pesticides just once, just in a single event. They may be repeatedly or chronically exposed to pesticides over the entire lifespan, life activity or nesting activity. And finally, and to summarize, we need a more holistic system-based approach to risk assessment. that includes several species, combinations of products, combinations of stressors, and realistic scenarios of exposure. That's all. I'd like to thank our research group at CREAF and at University of Bologna, and thanks to all of you for your attention.
Thank you, Jordi. Thank you very much. We actually have one question from the audience who wanted to know what are the active ingredients that you were testing. So what was the fungicide and the insecticide?
The insecticide was acetamiprid, and the fungicide was buconazole. So a neonicotinoid insecticide, one of the few neonicotinoids insecticides, is still allowed for use outdoors, for outdoors use in the European Union, and buconazole is an EBI fungicide.
Okay. And then there is also Andrew Quinn asking, what is the criteria that for you to select the pesticides that you're going to test in the combinations?
We tested this combination because we knew from previous laboratory work that the two compounds had synergistic effect. This is well known for certain families of fungicides and insecticides. Not all combinations do have synergistic effects. This is one that was likely to have synergistic effects. So that was one reason for choosing it. The other one is because both compounds are very commonly used in orchard crops and in a variety of other crops. Acetamiprid is actually one of the relatively low toxicity insecticide to bees. So it's being used a lot as a replacement of other nicotinoids that were banned because they were very toxic. So it's widely used, and the same thing with the buconazole. And the two compounds have been found together in bee matrices. So that's why I insist we're testing really realistic scenarios.
Mm-hmm. Thank you. So I propose, because your presentation raised more questions, but we need, so please, people, keep your questions. And so we still have 2 presentations to go. So we are going to focus on that. And then at the end, so if we have more time, please, so we will take them, okay? And otherwise, you can also write them in writing in the Q&A, part. So thank you very much, Jordi. So now I'm going to give the floor to, that was actually a transition toward impact of pesticides and risk or chemicals, let's say, on pollinators and risk assessment. So we are now going to have the, the presentation of Csaba Szentes from the EFSA, who is going to present the updates of the EFSA on the guidance document for the risk assessment of pesticides on bees. And Csaba is one of the ecotoxicologists in EFSA who have developed this guidance and who is doing risk assessment at European level. So with this, Csaba, I give you the floor.
Thank you very much, and thank you very much for the invitation. Can you confirm that you see my slides in the right? Probably not in the right.
We see your slides, but in the presenter mode, not in the.
Yes. So yeah, for sure. Of course, it happens. So I have to work out how to exchange. Probably I get rid of my second screen. Let's see what happens. Is it better? No.
No, still we are seeing the presenter mode now. No, wait, now it's your.
Okay, no, probably.
Now, yes, now.
Okay. Okay, then so I'm sorry again. Okay, so I'm going to talk about several projects which are going in EFSA. The first one is about a just finished project, is on the bee guidance document. And later on, I will talk about some ongoing projects. So some details on the review of the EFSA bee guidance document. Basically, it was mentioned in the previous presentation. So before going there, I would like to give a little bit of background about the history of the bee risk assessment schemes in the EU. First of all, I would like to mention the Sanco guidance document. So it's a commission guidance, which is based on an EPO guidance document. This risk assessment scheme is rather an incomplete, so it covers only honeybees, only basically at tier 1 at least, only the acute toxicity, only for spray applications, so rather incomplete risk assessment scheme. And in higher tier, some small scale studies, higher tier studies, field studies, for example, can be provided in the dossiers for the pesticide approval. Those small-scale studies are not necessarily robust enough to really dispatch effect if there is an effect. So this is a Sanco document. This is the currently official one. And this scheme is not linked to any protection goal. SPG stands for specific protection goal. So the wish from the risk managers, how much the bees should be protected against pesticide use. So this scheme is not linked to any of those. Then, in reaction to that incompleteness, let's say, EFSA has developed and published in 2013 its guidance document, which is more complete, more complex. It covers 3 types of bees, so all bee groups have some further options. Additional requirements was given for the higher tier studies, and it was linked to a level of protection which had been agreed that time. However, due to some criticism, mainly that it's too complex, the additional requirements are too harsh, so made the field studies unfeasible, as some stakeholders said, and they also complained about the severity of the protection goal, which had been considered at that time. This risk assessment scheme by EFSA have never been endorsed by the risk managers, and it is not fully in use. Therefore, this triggered the wish to update, to review this guidance document, and that is the project I'm trying to talk about. So, the review project of the 2013 guidance started in May 2019 and lasted 4 years. It was published May this year. The upper part of the slide is more about the scientific part. So there had been during this 4 years, there was a lot of data collection, intensive development of different risk assessment methods. Meantime, there was a lot of stakeholder consultation, also public consultations. The project had also a less scientific, more political dimension, and this is the specific protection goal setting. In the next slide, I will give some details on the specific protection goal. So this is the protection goal setting is more in the hand of the risk manager. So it's not it's basically then the risk managers express how much they want to protect the bees against pesticide use. But however, they want, so it's a political decision. Nevertheless, they want to have an informed decision, so they need data and the interpretation of data. And that was the part where the EFSA working group has helped them. So you see the different steps of this journey. So first, some approaches, the potential approaches, how to reconsider the specific protection goal setting was provided to them in a document. Then they selected one of the 4 approaches. That approach was further elaborated based on the selected methods. In the next step, several relevant data had been collected, and based on the approach they selected and the data supporting this approach, risk managers were able to agree on the new SPG at very high level. So at Agrifish Council, which means the ministerial level, they agreed that for honeybees, the specific protection goal should be 10%. In the next slide, I will give you some more details. Then the EFSA working group also collected some data for white bees, so bumblebees and solitary bees. Our decision on the threshold, so similar than the 10% here, could not be done by the risk managers because they thought that the data what is available is too little to make an informed decision. Then the details about how the specific protection goal looks like, as said by the risk managers. So this is the 10% here for honeybees, which is the magnitude. So how much bees should be protected, and what to protect is the attribute, so colony strength. So what does it mean? That the maximum tolerable impact on a honeybee colony due to a pesticide use is not more than 10% decrease in the colony strength, which is the number of adult bees in the hive. As mentioned, for bumblebee solitary bees, similar threshold could not be agreed due to the too few data. Nevertheless, we know the attribute to be protected. So this is the colony strength again for bumblebees and the population abundance for solitary bees. So to know this, the agreement on this is already an important, because in future field studies or higher tier studies in general, semi-field or field studies, we know that which is the end point which should be in the focus. This is the colony strength and the population abundance. Lastly, about this project, I go back again to the scientific part. The new guidance document really made some very new proposals in methodological, it means on methodology. There is a long list of new approaches, innovations. I'm highlighting here only three of those. So the hazard characterization is moved away from point estimation to considering the full dose response. So in the past, or even in the present, from the hazard, so just imagine a laboratory study, the risk assessment normally use a point estimate, which is, for example, an LD50. However, especially if we are in the around in the lower part of the dose response, the shape of the dose response curve does really matter. And now the working group worked out the methodology to work with this shape. So we consider the full dose response. Then the risk assessment combines the risk cases. For bees, we require a full data package from the laboratory level. So we have, for honeybees, we always have in the dossier acute contact test, an acute dietary or acute oral test, a chronic oral test, and the larva test. So these are the basis of the tier 1 risk assessment. And in the past, even in the present, that meant 4 different risk assessments. So each were individually considered. So there was the hazard, which was a point estimate in the past. Now it is a full dose response, an exposure estimation for each, and they were considered individually. Now the working group developed a method to combine these 4 and give a prediction immediately for the colony in case of honeybees. So the 4 will be combined, and there is a prediction immediately to the colony, so a percentage, which compares directly to the SPG. Then a statistical paradigm change. This is mainly for the higher tier studies, like semi-field or field studies, mainly field studies. In the present, in general, and not only for bees, but really generally, we use the so-called difference test. The difference test, so first of all, I'm not talking about a new statistical method. The method statistical methods are still the same, the approach is different. So the difference test as approach, looking for differences between the control and the treated field. So we have a treated field with the pesticide and the control field, and we compare, for example, the colony strengths. And the conventional statistical approach, looking for differences between these two. If there is no difference between the two, then it very likely lead to low risk conclusion. However, whether a field test is good enough to dispatch effect, also the study design, the size of the test, the robustness of the study has a very important role. So in a weaker study, you may not see differences, but it does not necessarily mean there is no effect. And that's why the working group decided to basically make an upside down and ask for the demonstration of equivalence. So in the future, the applicants has to demonstrate that the control and the treated are equivalent. And this automatically includes the study design. So only for those ones which are risky, which are more more risky, only equivalence can be demonstrated only with robust enough studies. So with that, I'm finishing talk about the bee guidance document, and I'm mentioning 3 additional projects which are ongoing in EFSA, APISRAM, EANEAS, and PERA.
Okay, Csaba, I just need you to speed up a little bit, okay?
Okay, sorry, I don't see the clock, but okay. Okay, the APISRAM is a honeybee colony effect model for pesticide risk assessment and more. Then I just very quickly I mentioned that there are different versions. Now we are working on version number 3, which will be implemented next year and finalized in 2025, ready for use in risk assessment for pesticides. Just quickly mention, it has different modules, not only pesticide module, also other stressors will be considered. Then ANAEAS, which is advancing the environmental risk assessment on non-target arthropods for plant protection products by accounting for impact of ecosystem services and ecological functions. So this is a kind of setting the preparation for guidance development for non-target arthropods. It has 3 main aims, which will help to establish so that kick off basically the guidance document development, and also the risk management discussion on the protection goal for non-target arthropods, including pollinators, of course. The last one is PERA, which is a very wide project on terrestrial, mainly terrestrial organisms. It's some of the sub-project, and I will speed up. Some of the sub-project are ready to start next year, at least awarded, so we can do it. We really want a full description on the agricultural landscape with this work package 1. Then more definition will be set for so-called low concern pesticides. Then there are areas on development for the exposure characterization, the hazard characterization. Then other new methodologies will be investigated to be used in the future for the risk assessment, and the common platform is also in the horizon. Okay, this is my very last slide, where I just would like to advertise that we are looking for stakeholders. So you still have, if some of you are interested, a few days to apply for a stakeholder for environmental risk assessment. This is mainly for the future development of guidance document for non-target arthropods. Thank you very much, and sorry for the time.
Oh, thank you, Csaba. No, that's good. We actually have one question from the audience that is from Michael Heyer, saying, thanks for the update. Why is the specific protection goal colony strength instead of fitness parameters?
Well, the colony strength is a fitness parameter, isn't it? So if the colony is strong enough, then it also translates. It also means it's fit. I'm not sure what fitness parameter exactly means otherwise.
Yeah, I invite Michael to say something else about this and to clarify his question. Meanwhile, so there is another one, and then with this we follow to Jaana. So the bee guidance exclusively refers to adult honeybees and honeybee larvae in the test. The central element in a bee colony is the queen bee, and unfortunately, drones are completely overlooked. Drones mate with a virgin queen. Studies have shown that the fertility of human sperm has decreased by 50% due to pesticides. The vitality of drone sperm is crucial for wild and honeybees. The impact of pesticides on sperm must be taken into account in the future. So it's more a statement than... So we can make it a question. Will the impact on the pesticide on sperm be taken into consideration in the future risk assessment?
In the future, everything can happen. So we are aware that there are some studies on the sperm, for example. In the future, of course, if it is really demonstrated by the scientific community that it's a key point, then we can consider, of course. What I can say for today, that at least considering some biological, the important biological parameter like the food consumption of the drones versus the workers and the body size, etc. We consider that they are generally covered. From this point of view, also the queens are generally covered from there are some biological argumentation behind this. Nevertheless, the sperm is really so that the workers do not have it. So yes.
Thank you. So, well, Michael asked in the chat, so I ask you to please answer in the question and answers. Okay. And with this, I give the floor to our last intervener, who is Jaana Laitinen. I don't know if I pronounce it correctly. Thank you very much, Csaba. So Jaana, is a scientific officer at the Biocide Active Substance Unit in the European Chemical Agency. She was involved in the development of the last week published, if I remember well, guidance document for the risk assessment of biocides on bees. And so she is going to present us this guidance. So Jaana, the floor is yours.
Thank you very much. And thank you for the invitation. I will wait that I see the slides. If you can share them.
Arriving, just a minute.
Yeah, so in the presentation, I will present the work by the expert group for this new ECHA guidance on the risk assessment of bees. And then also the status of the non-bee pollinators. So next slide, please. Indeed, I will explain about the background and the development process, and I will give an overview of the new ECHA bee guidance. And for the non-bee pollinators, I will say some words about the status and approach. So as a background, as for EFSA, also ECHA has received a mandate from the Commission to develop a guidance for the risk assessment of arthropod pollinators, including bees. So in other words, the request was not restricted only to bees, but we were asked to develop guidance also for the so-called non-bee pollinators. And another important aspect was that we were asked to take into account the available guidance from EFSA for the plant protection products. Next slide. So to meet the requirements of the mandate, an ECHA expert group was formed, and we have 21 experts from different member states. And also in order to ensure the harmonization with the pesticides work, also experts from EFSA have been involved in the expert group. Next slide. So looking back to the development process, after receiving the mandate in 2019 and after forming the expert group, in the initial step of the development process, the expert group has created a so-called scoping document. This was important since for biocides, so far we didn't have any guidance for the risk assessment of bees. So the work started by looking into the relevant exposure routes, relevant biocide active substances and products, and so on. And for that, there is a standalone document published. Then started last year, end of last year, the drafting, actual drafting of the guidance was started, which was then finalized in beginning of this year. And of course, for us to finalize our guidance, we needed to wait for the finalization of the EFSA guidance for the pesticides to be able to harmonize the guidance documents. Then after that, we have run a number of consultation steps for the scientific and technical aspects of the guidance, and also with the competent authorities for biocides in the autumn this year. And as we have already heard today, last week, there was the Biocidal Products Committee meeting last week, which has adopted this opinion for the Commission mandate. So this means that we have finalized the final formal step of the development process. But for the publication, we still need a little bit more time to work on the editorial aspects of the guidance. And that is expected beginning of next year. Next slide, please. Thank you. So here we present the overview of the ECHA guidance document for biocides. The structure is very similar to the one for the pesticides, of course. And in our guidance, we focus on the biocide-specific parts. And for the description of the detailed methodology and scientific background, we refer back to the documentation available from EFSA. But as mentioned, there are many similarities between the guidance for biocides and pesticides. For example, the lower tier effects assessment and risk assessment is same or very similar. But since we have most of the differences are related to the exposure from biocide and pesticides use, therefore the exposure assessment chapter is the one that is like a very specific to biocides and different from the pesticide guidance. We also have implemented the time reinforced toxicity and sublethal effect assessment with this new type of assessment for biocides. And in addition, we have instructions for other assessment like the metabolites and mixtures. And finally, we have recommendations for future research and development. Next slide, please. So the expert group has concluded that indeed it's possible to transfer the assessment framework proposed for pesticides also to the biocide assessment. For example, the specific protection goal for honeybees of 10% for the colony strength has been taken over. Also, the standard testing methods for toxicity bees are applicable to biocides, but then especially related to exposure calculation, certain adaptations are needed due to the differences in the application methods and also some data availability for biocides. And regarding the higher tier assessment, this is not directly suitable for biocide, since, for example, the field studies doesn't really represent the biocide use. Next slide, please. So here to show that the risk assessment scheme for biocide is very similar as in the revised EFSA guidance for pesticides, but the part that are specific to biocides are indeed related to the questions when a risk assessment is relevant for biocides, what are the relevant exposure routes, and also the higher tier risk assessment part. Next slide, please. Also to shortly mention that in the guidance, we cover the oral exposure via the contaminated pollen and nectar, and also contact exposure directly or via contaminated matrices. Next slide, thank you. So then looking a bit more in detail to the exposure assessment, which is then specific for biocides. As you may know, biocides are always approved under a specific product type, and in total there are 22 different product types, and these cover a number of different kind of exposure routes and types of application. And this has been one of the key questions along the guidance development. So the expert group has screened through all the different kind of exposure scenarios that are available for biocides. And finally, it was concluded that the assessment is focusing on the product type 18, which are insecticides for the control of arthropods. But also to mention that it doesn't exclude that in the future there can be also other uses or product types for which we will need a risk assessment of bees. For example, at the moment, the expert group is looking into the product type 19, which is covering the repellents and attractants. Then still for the product type 18, yes, next slide, please. Thank you. It covers a number of uses, but it was concluded that from those numerous uses under PT18, there are 4 main sources of exposure that are relevant for the bee risk assessment. And therefore, in the guidance, we provide instructions how to perform exposure and risk assessment for these 4 uses: for the application of manure and sludge, spraying of walls and foundations of buildings, irrigation of private gardens, and also the large-scale spraying. Next slide, thank you. So then a few words for the non-bee pollinators. So as part of the work done on the mandate, the expert group has conducted a literature and database search for the basic ecology, biodiversity, and sensitivity to biocide of the non-bee pollinators. And as a result of that exercise, we have identified some key data gaps. Namely the lack of standard test guidelines, lack of information on the basic ecology and biology of the pollinators, also lack of information for the most relevant route of exposure and life stages, and also there is a lack of commercially available test species at the moment. So due to these identified data gaps, it was not possible to propose a risk assessment scheme at the moment. But in any case, the expert group has considered that these non-bee pollinators will be covered under the risk assessment of non-target terrestrial arthropods. And then this means that there is an update required for the already existing biocide guidance documents for the terrestrial risk assessment. So in order to complete this part of the work, of course, the outcome and results from the relevant scientific projects will be important for the future developments. So to summarize, we now have soon available the new ECHA bee guidance for the risk assessment of bees, which is taking into account the guidance on pesticides. And this is expected beginning of next year. And in the first version, the focus is on the insecticidal biocides, where we have identified significant exposure to bees. And for the non-bee pollinators, further research will be needed to define a quantitative risk assessment method. That was my last slide, so thank you very much.
Thank you, Jaana. We are a little bit overdue by the time, but it's fine. We can have these 5 academic minutes, let's say. We have a question in the chat that is, but I don't know, maybe you have already answered because you explained it extensively. So what are some of the identified exposure scenarios of bees to pesticides that you have explored, I guess, for this type 18?
Sorry, I think I didn't get. What are the relevant exposure
scenarios of bees?
From the biocides.
From the biocides, yeah. So I guess, so you already explained the different type of biocides that exist. So you have focused on 2 mainly.
Yes, yes.
So from these 2, what kind of exposure would you expect? But I think you have already said.
Yes, yeah, I think this was mentioned that we have these 4 main sources of exposure when if there is manure or sludge that is contaminated, and then the plants, the nectar or pollen is contaminated by the plant, and then the spraying of the buildings, use of contaminated irrigation water, and then the large-scale outdoor spraying.
Mm-hmm. And so then I add my own question. Normally for the pesticide guidance document, there is a procedure afterwards for member states to embrace it, let's say, in order to have the guidance document as their reference methodology for risk assessment. Is this the same for the biocides? Will you still have to pass by the standing committee meeting, so the member states of the European Union embrace your methodology for biocide risk assessment?
Yeah, thanks. That's a good question. We don't have that kind of process, but actually we always have a date of applicability for our guidance, and that date hasn't yet been agreed for our bee guidance, and that is still under discussion. But we expect to have a conclusion for that by end of this year or beginning of next year. So then we will know by when the new guidance will start to apply for active substances and then for biocidal products.
Great, thank you. I'm curious how for similar products, let's say, there are different procedures at the different level. It's curious to see this incoherency somehow, no? Because someone would expect that the procedure is follow, like the follow procedure is the same for everything. But okay, thank you very much, Jaana. So we don't have any more question and answers, and we have 5 minutes above the schedule, let's say, finish our time. So with this, today we learned a lot of activities that we are having at European level, bringing us a lot of new data about the trends and the richness, let's say, about and the abundance of pollinators, wild pollinators in Europe. that will help us hopefully have indicators about how efficient all the policy measures are in practice. So that is very, very nice. We also learn about the project on good beekeeping practices. So we hope that this information and training can get to all beekeepers, and that the beekeepers can actually co-create, because there is a lot of knowledge out there in the field of good beekeeping practices, so it can be spread. And for the risk assessment of chemicals on wild pollinators, and actually also in honeybees, we learned today a lot about what is done in the research, let's say, methodologies that are being developed every day, and that eventually could be, so in the future, could be integrated into the risk assessment. So the researchers out there who are developing this kind of methodologies, please keep up to that, publish them, and very important, transfer your publications to the public that eventually could embrace them. So this is actually the European agencies that are responsible for developing methodology of risk assessment, because a lot of things can happen in the future, and the methodologies can always improve. With this, I would like to thank you very much for your attention, for your time today, and we hope you will be following the other events at the European Pollinator Week. Thank you very much. Good afternoon. Bye.